Revision of experiment number 12, histology of human organs, since I didn't use a printed handout when I first explained it to you in class. So I hope this proves to be useful for your exam. First of all, we need to know that histology is the study of human organs. We need to know this definition. And in this experiment, we're going to discuss 6 different organs in detail. First of all, we're going to begin with human skin. We need to know that it's the largest organ in the integumentary system. The integumentary system, we don't need to know what it is actually, but just for clarification, it's the skin and all the appendages that are around it, such as hair and nails, and the hypodermis as well, which act to protect the body from various kinds of damage. So that's the integumentary system. As if we recall from class, the hypodermis is a part of the integumentary system, but it's not part of the skin. So the skin and the hypodermis and the nails and the hair are all a part of the integumentary system, but we don't need to know this. This is just for clarification. We also need to know that the human skin has multiple layers of ectodermal tissue. By ectodermal, we mean that the human skin cells originate from the ectoderm, which is one of the 3 primary germ layers in the very early embryo. So as you can see here, the ectoderm gives rise to epidermis skin cells. We don't need to know this as well, just that the ectodermal tissue is found in the human skin. So we need to know what the 2 types of human skin are. We have hairy skin, which is found everywhere on the body where we have hair follicles, and glabrous skin, which is areas of the skin in which hair does not grow, such as the skin of our lips, the palms of our hands, and the soles of our feet. Now, we're going to talk about the skin itself. We have 2 primary layers of skin, the epidermis and the dermis. First of all, we need to know that the epidermis consists of stratified squamous epithelium with underlying basal lamina. The other term for basal lamina is basement membrane. The epidermis is the outermost layer of the skin, and it serves as a natural barrier to infection. We also need to know that it's waterproof and that it's avascular, meaning that it does not have blood vessels. The epidermis itself does not have blood vessels, so the deepest cells are nourished from mostly by diffused oxygen from the surrounding air, and to a far lesser degree by blood capillaries coming from the basement membrane or the basal lamina. We need to know that the skin can be divided into 5 sublayers, starting with the stratum corneum, the stratum lucidum, the stratum granulosum, the stratum spinosum, and the stratum germinativum, which is also known as stratum basal. The outermost layer is the stratum corneum, and the innermost layer is the stratum basal. Now we need to know that these first 2 layers, the corneum and the lucidum, they're only found in the palms of our hands and the soles of our feet. So the only parts of our skin that have all 5 layers are the palms of our hands and soles of our feet, because they need extra protection from friction. All our other skin starts from the stratum granulosum. So here we can see the layers of the skin. As we can see here, we have the stratum basal, then the stratum spinosum and granulosum. Then we have the lucidum and corneum. As we can see here, the last 3 layers, granulosum, lucidum, and corneum, we can see that the cells have effectively become keratinized, meaning that they've lost their cytoplasm and they've lost their nucleus due to insertion of keratin. We're going to discuss why that's important later on in this video. Moving on to the dermis, which is the second layer of the skin, we need to know that, in contrast to the epidermis, which consists of stratified squamous epithelium, the dermis consists of connective tissue. It's basically the layer of the skin beneath the epidermis, it's tightly connected to the epidermis via the basement membrane or the basal lamina. We need to know that it's neural, meaning that it has many nerve endings that provide sense of heat and touch, and that it's vascular. It has blood vessels. Here, the epidermis is avascular. It does not have blood vessels, but the dermis itself is vascular. It has blood vessels. We also need to know that the dermis contains hair follicles, sweat glands, sebaceous glands, apocrine glands, lymphatic vessels, and blood vessels, and this is what gives the dermis its vascularity, the blood vessels. The dermis can be divided into 2 areas, the papillary region and the reticular region. Now, the papillary region it consists of the finger-like projections that are found in the dermis. So these finger-like projections are known as the papillary layer. And this part here, under the finger-like projections, is the reticular layer. The papillary region consists of areolar or loose connective tissue. As you can see here, it's finger-like projections, and it provides the dermis with a bumpy surface. The reason or the function of the papillary region is to strengthen the connection between the dermis and the epidermis. So that's the function of the papillary region. The reticular region consists of dense, irregular connective tissue. As we can see here, this is the reticulary region. Its name comes from the fibers it contains. So it's called reticular because it contains collagenous, elastic, and reticular fibers. These protein fibers give the dermis strength, elasticity, as well as extensibility. The dermis, or the reticular layer, contains sebaceous glands which produce sebum, which is a naturally healthy skin lubricant. We need to know also that if excessive sebum is produced or there is an overproduction of sebum, the skin becomes thick and heavy in texture. As we can see here, or this is before, first of all, how are cells formed in the epidermis? First of all, in the stratum basale, which is right here, we have mitosis, which leads to daughter cells. The daughter cells begin to move up the stratum, going from the stratum, starting from the stratum basale and moving upwards through this spinosum and granulosum and lucidum and corneum, if we're talking about the palms of the hands and soles of the feet. And as they move away from the stratum basale, they die as they do so. Why? Because they become isolated from their blood source. The blood reaches the cells through the basement membrane, through this lining here that separates the epidermis from the dermis. So as the cells are moving away from this basement membrane, they become cut off from their blood source and they therefore die. This movement up the various strata causes cells to release their cytoplasm and their nucleus due to insertion of keratin. So here we also have to know that the cells lose their nucleus as well. As we can see here, the cells of the upper strata lack both the nucleus, and we can't tell if they lack a cytoplasm, but the most obvious thing here is that they do not have any nuclei. So if we have a question in the exam which says, which has an arrow pointed here and says which types of cells are these, we can easily tell that they're keratinized cells because they lack a nucleus. So this movement causes cells to release their cytoplasm due to insertion of keratin, and this process is called keratinization. When the cells eventually reach the stratum corneum, they slough off, meaning they are rubbed or they fall off. Now, what is the function of the keratinized layer of the skin? What is the function of this layer right here, the keratinized layer? It keeps water inside the body. So it keeps water here, it prevents it from escaping, and it keeps harmful chemicals and pathogens out. So it doesn't allow harmful chemicals and pathogens to enter, and it doesn't allow water to leave. So that's the function of the keratinized layer. As we mentioned before, pathogens are microorganisms that have potential to cause disease in humans. Next question. What is the function of blood vessels in the dermis? The blood vessels that are found in the dermal layer, what is their function? They have their function is twofold: to provide nourishment and waste removal from its own cells as well as the cells of the epidermis. So it provides nutrients for the dermal and epidermal cells, and it removes waste from the dermal and epidermal cells. It does this with the epidermal cells through the basement membrane. Next, the final part of the human skin is the hypodermis. Now, as I mentioned before, the hypodermis is considered to be a part of the integumentary system. The hypodermis is right here, the subcutis. The hypodermis is part of the integumentary system as a whole. However, it's not part of the skin. The skin is simply the epidermis plus the dermis. The hypodermis is not a part of the skin. However, it is a part of the integumentary system. So this is all we need to know about the hypodermis. It's not part of the skin, and that it's found directly underneath the dermis. We have to know that it contains 50% of body fat. So 50% of body fat in a normal sized individual will be found as subcutaneous fat, as in fat in their hypodermis. And we need to know that hypodermis has two functions. It attaches the skin. By skin here, I mean the dermis and the epidermis. It attaches the skin to the underlying bone and muscle, and it supplies the skin with the blood vessels and nerves. So this is the dual function of the hypodermis. But remember, the hypodermis is not a part of the skin. It's part of the integumentary system. The skin is the epidermis plus the dermis. With this, we finished human skin. The next organ we're going to talk about the next organs we're going to talk about are the human intestines. The basic function of the human intestines is to digest food, to enable the nutrients released from food to enter the bloodstream. We have two major subdivisions of the intestines. The small intestines, which are much smaller in diameter but longer and larger than the large intestine, and their function is to digest food and absorb nutrients. And we have the large intestines, which have a larger diameter but they're overall shorter, and their main function is absorption of water. Now the intestinal wall, as we can see here, this is a picture of the intestine. As we can see here, the small intestine has a smaller diameter, but it's much longer than the large intestine, which has a larger diameter, but it's shorter. Now we need to talk about the layers of the intestinal wall. We need to know that it's divided into 4 layers, and the manual also mentions that these 4 layers are seen in the jejunum and the duodenum. First of all, we have the mucosa, the mucosal layer. The mucosa, we need to know that it contains the villi, which are finger-like projections lined with simple columnar epithelial tissue. We need to know this fact. It might come as a question in exam. What type of tissue are the villi made of? In this case, it would be simple columnar epithelial tissue. The manual also mentions that simple columnar epithelial tissue have an ovoid nucleus at their base, and we already know this fact from the previous experiment in animal tissues, that simple columnar cells have nuclei all at the same height at their base. And the mucosa also contains goblet cells. The function of goblet cells is to secrete mucus, and this mucus prevents friction from food passing through the lumen. We mentioned that lumen refers to any hollow area inside a tube. So the lumen of the small intestines is the hollow area. We need to know that the surface of the epithelium has a striated border. Striated means it appears in lines due to the presence of the microvilli, and this striated border increases its absorptive surface. In the mucosa, we have a basement membrane, which is, the manual says, it's made of reticular fibers embedded in an amorphous protein polysaccharide ground substance. So the reticular fibers are found inside this non-living substance. If you remember from the animal tissue lab, we said the ground substance is the non-living part of connective tissue. But here we don't need to know that. It's not referring to connective tissue, but ground substance refers to a non-living thing. We also have in the mucosa capillaries of the villus that form a network under the basement membrane. And we have epithelial cells of mucosa that absorb water and digested food and transfer them to blood in nearby capillaries. Finally, we have the central lacteal, which is a lymph vessel located near the villus and becomes distended after absorption of fat. We have to know that it contains simple tubular intestinal glands, the crypts of Lieberkühn, and the lymphatic nodules. So all of this is in the mucosa. So far, we've only discussed the epithelial layer of the mucosa, and that by epithelial we mean the simple columnar epithelial tissue. But we also need to know about the lamina propria. The lamina propria is the region under the epithelium, and it is the part of the mucosa involving the core of the villus. We have to know that it is composed of loose connective tissue. So this part is simple columnar epithelial tissue. This part is loose connective tissue or areolar connective tissue. It's also, in addition to loose connective tissue, it has blood vessels, a lymphatic vessel, smooth muscle fibers, and other cells of the connective tissue. The lamina propria ends at the edge of the muscularis mucosa, which is composed of a band of smooth muscle fibers and is a few layers thick. So all of these 3 layers, the epithelium, which is made up of simple columnar epithelial cells, the lamina propria, which is made up of loose connective tissue in addition to other things, and the muscularis mucosa, which is made of smooth muscle fibers. These are all a part of the mucosa. Next, we have the submucosa here. It is composed of loose connective tissue. And we need to know that the duodenum, but not the jejunum, the submucosa of the duodenum, but not the jejunum, contains compound tubular mucus glands of Brunner. We also have to know that this submucosa is a continuation of the pyloric glands found in the stomach. This is a piece of information from the manual. So this is the submucosa. Next, we have the muscularis. The muscularis contains two layers of muscle. The inner layer of muscle in the muscularis is circular muscle, and the outer layer of muscle in the muscularis is longitudinal muscle. So the muscularis contains inner circular and outer longitudinal layers of smooth muscle fibers, and these layers of muscles allow the intestines to move. These inner and outer layers of muscle are separated by reticular and collagenous connective tissue containing nerve fibers and parasympathetic ganglion cells. And we need to know that examples of intestinal movement include peristalsis and segmentation. Now, the basic difference between peristalsis and segmentation is that peristalsis is controlled by the longitudinal fibers. So this part here controls peristalsis, and this part here controls segmentation. But we don't need to know this for the exam. So here we have a diagram of the peristalsis. As we said, it's most famously seen in the esophagus during swallowing. So this is an example of peristalsis. The outer layer the outermost layer of the intestinal wall is the serosa. So here we can see it says serosa or adventitia, and we'll get to that in a moment. We need to know that the serosa consists of loose connective tissue and mesothelium. Mesothelium is simple squamous epithelial tissue. When the serosa covers the intraperitoneal organs, it is called serosa, and when the outer layer, this layer, covers the retroperitoneal organs, it's called adventitia. What are intraperitoneal and retroperitoneal? Basically, intraperitoneal organs are those bound to the posterior abdominal wall, and retroperitoneal organs are those not bound to the posterior abdominal wall. So when it's not bound, it's retroperitoneal. It is called adventitia, and the main difference between the two is that adventitia does not have this mesothelium. It's only composed of loose connective tissue. But the serosa covering the intraperitoneal tissue consists of loose connective tissue as well as mesothelium. We need to know that most of the duodenum is retroperitoneal, meaning that if we go to the intestinal wall of the duodenum, its outermost layer is going to be called adventitia, but the entire jejunum is intraperitoneal, meaning if we go to the intestinal wall of the jejunum, its outermost layer is going to be serosa. We discussed the lamina propria and muscularis mucosa beforehand, and this basically covers the human digestive system. After the human digestive system, we have the human kidney. The human kidney consists of two bean-shaped organs, as we can see here, and it's covered with the renal capsule, which is tough and fibrous and consists of two layers. The inner layer, which is made up of myofibroblast cells, and the outer layer, we're going to discuss that later. Resting on top of each kidney, at the top of each kidney, we have adrenal glands. And the space between the cortex and the medulla, right here, the space between the cortex and the medulla, consists of two layers of fat, consists of blood vessels running parallel to the surface of the cortex that form an arcade shape. Each kidney and this adrenal gland, so this whole structure right here, the kidney plus the adrenal gland, is surrounded by two layers of fat, and these layers of fat are perirenal and pararenal, and as well as something called renal fascia. We don't need to know what these are, we just need to know that these layers surround both the adrenal gland and the kidney. The material present between the main parts of the kidney is formed of blood vessels, as we mentioned before, blood vessels here, stroma and collecting tubules. So this part between the cortex and the medulla is composed of blood vessels, stroma, and collecting tubules. These appear as a colloidal substance altogether. So we just need to know this part because it's in the manual. Now we have the nephron. What is the nephron? The nephron is that was a mistake. The nephron is basically the functional unit of the kidney. It consists of the renal corpuscle plus the renal tubule. So here we have the renal corpuscle plus all of this right here. These three layers are referred to as the renal tubule. So all of this right here is the nephron. So what are the functions of the nephron? Filtration, reabsorption, and secretion. As we said, the nephron is the functional unit of the kidney. We need to know that there are around 1 million nephrons in each kidney. So this thing right here, this whole structure, we have almost 1 million of in each kidney. The function of nephrons are to regulate blood pressure and volume. Or excuse me, the nephrons that regulate blood pressure and volume are controlled by the endocrine system. So now we're going to discuss the parts of the nephron, which are the corpuscle and tubule. The corpuscle is basically made up of a glomerulus, right here, as well as the Bowman's capsule. So the corpuscle is made up of the glomerulus, which is a network of capillaries, and a cup-like sac known as the Bowman's capsule. So this is the corpuscle. The tubule is a specialized tubular structure made up of the proximal convoluted tubule, the loop of Henle, and the distal convoluted tubule. So the proximal convoluted tubule, the loop of Henle, and the distal convoluted tubule, these are all referred to as the renal tubule. What are the hormones that help in the regulation of the functioning of nephrons? Aldosterone, parathyroid hormone, and antidiuretic hormone. So these hormones help regulate the functions of nephrons. We need to know what the Bowman's capsule is made of. The Bowman's capsule is made of an outer, or another word for outer, parietal layer made of simple squamous epithelium. So the outer lining of the Bowman's capsule is made up of simple squamous epithelium. So this lining is simple squamous epithelium. What is the loop of Henle made of? It is made up of the descending thick limb, the thin descending limb, the ascending limb, and the ascending thick ascending limb. In some books, it just refers to the ascending and descending thin limb as just the thin limb. So here we have descending thick limb, descending thin limb, ascending thin limb, and ascending thick limb. Furthermore, we need to know these tubular structures. What are they made of? The thin limb is made up of simple squamous epithelium. But all other tubular structures are made up of simple cuboidal epithelium. We need to know this because it's in the manual. This thin limb is made up of what? Simple squamous epithelium. But the other limbs and tubular structures are all made up of simple cuboidal epithelium. How does filtration occur? Filtration first occurs in the renal corpuscle, in this part. That's where the initial step of filtration occurs. Water and ions are forced into the Bowman's capsule. So they go from here and they're forced into the Bowman's capsule. And then the filtrate passes into the renal tubule. The renal tubule, as you might remember, is all these three parts together. The brown part and the green part and the pink part are all referred to as the renal tubule. So this is basically how filtration occurs, but we need to know that it's initiated, or it first happens in the renal corpuscle, where the water and ions move from the glomerulus into the Bowman's capsule. Now, what is the significance of the afferent and efferent arterioles? You might have noticed while you were memorizing the diagrams that we need to know something known as the afferent arterioles and the efferent arterioles. Basically, the glomerulus comes from the afferent arterioles. So it arises from the afferent arterioles, but it leaves the renal corpuscle as efferent arterioles. So this part afferent, this part efferent. If you might remember from the nerves that we talked about in the animal tissue lab, afferent means coming towards and efferent means going away from. So afferent, they're coming towards the corpuscle. Efferent, they're leaving the corpuscle, going away from. Another thing we need to know about the efferent arterioles is that the peritubular capillaries, which surround the renal tubules, which are found everywhere, these peritubular capillaries, they're found everywhere around the tubule. So this part here is all surrounded by peritubular capillaries. They arise from efferent arterioles, which go away from. Next, in the kidney here, I put the diagram that's in your manual, along with extra information that we need to know. Here we can see, first of all, this is the same diagram as the one found in your manual, along with the information that's needed. First of all, the renal cortex is defined as the region that contains the renal corpuscles. So this part here is found in where? This part. It's found in the renal cortex. So the region that contains this corpuscle is known as the cortex. It's the pale outer portion of the kidney, and we need to know that it's composed of two types of tissue, the medullary rays and the labyrinth of Ludwig. And we need to know that the labyrinth of Ludwig in particular contains the glomeruli. What are the glomeruli? It's this part. So this part here is found in the labyrinth of Ludwig. This whole part is found in the cortex.